Okay, so this is where we left off. We're talking about the phase of the gastric activity. So we said that there's cephalic, gastric phase, the intestinal phase. And let's make sure I unmuted myself because I did mute earlier. Excellent. So the reason why that shouldn't look good. Okay, so let's take a look at the cephalic phase first. So the cephalic phase is obviously the cephalus, which is the head, right? This is basically kind of like a preliminary sort of a priming of the pump phase, if you will. And so typically this is triggered by your hunger and your sense of craving and stuff like that, right? So like if you smell food cooking, if you see it, you taste it, kind of gets that whole sort of mouth watering thing going. So there's kind of like that preliminary phase. It's like your body is preparing in anticipation that you're going to eat something, right? And so part of the reason why you salivate is because your body's preparing for the food. So you're already upregulating your salivary glands because you're anticipating having to chew stuff. And of course, part of that is mixing it with your saliva. And so that information then, as you're sort of taking all that in, smelling food, tasting it, seeing it, thinking about it even, obviously your perception of food has a lot to do with your limbic system, right? One of the reasons why food has several, not just one, but several of its own networks that does quite well, all of them. It seems to be an inexhaustible source of programming. We'll always watch a good food show, even if it's ridiculous, like Is It Cake, which is fun, but completely useless. It's actually fascinating because I always get it wrong. I'm like, seriously, really? Okay, anyway, I totally like in awe of what they can do with that sort of stuff. But anyway, there's often a lot of emotional charge associated with that, right? Because your sense of smell, if you're smelling food, can also be a repulsion, right? So it can do just the opposite to you. And so all of that information emotionally is going down through your vagus nerve. And this is where your vagus nerve does that sort of delegation of coordination. Remember we talked about the central nervous system sort of delegates all the digestive stuff to the enteric nervous system. And this is where this is going. So this is the enteric nervous system. So the vagus nerve will hand it off to a couple of plexuses that are associated with that enteric nervous system. The first one we're going to see here, which is the submucosal plexus. Notice it's the submucosa. That's what the term submucosal comes from. So you're talking about the submucosa. It's a plexus that's going to be basically in the stomach cell wall. And the submucosal plexus has a couple of activities. First of all, one of the activities that it has in response to your smelling food or thinking about food, you're kind of getting hungry and that sort of thing, is one of the things it'll activate the mucus cells in the mucosal liner of the stomach. So you're going to start to upregulate mucus production. That's basically one of the things you're going to be doing. You also stimulate the chief cells. Remember, the chief cells are the ones making pepsinogen because you're anticipating eating food, a lot of which is probably going to be protein. So you're already starting to fire up the engines of creation of pepsinogen so you can get a head start on that protein digestion. And of course, the parietal cells, which we talked about before, were the cells whose job it is to make the stomach acids or the hydrochloric acid. So all these guys are getting upregulated in response to the submucosal plexus because you're smelling food. Okay, so that's basically what's happening. So it's kind of like you're priming the pump, you're getting your stomach ready and your gastric juices ready to take in food. The other thing it also does is it also stimulates G cells. Remember, we talked about G cells very briefly, and we said the G cells are basically in the pyloric region of the stomach. They're deep down in the gastric pit, and their job is to make gastrin, which basically stimulates all of these. So gastrin is the hormonal component of the activation and stimulation of the stomach. So this stimulates gastric activities. So it's kind of like a little bit of a positive feedback loop, kind of feeds back on itself and reinforces those gastric activities, which then makes more gastrin, which then feeds back and increases it. So you kind of get this sort of vicious cycle until somebody puts a stop to it. So that's your cephalic phase. You're getting yourself ready. Then you move on to the next phase, which is the gastric phase. So at this particular point now, what's happened is you've actually eaten. So you've swallowed some food. And so what's happening here is a couple of things. So first of all, let's start off with our regulation. Let's start off with our submucosal plexus, which is still firing. So notice you're still activating your mucus cells. So you're still actively making mucus even into the gastric phase. Your chief cells are still very busy making pepsinogen, because now you're kind of like at the apex of your gastric activities. Parietal cells making lots of hydrochloric acid. Just basically, the three of these sort of represent your gastric juices. So these are basically what you vomit up. Now, lovely. I always think of it in terms of that. So very unpleasant place to be. Don't ever get into the business end of somebody's stomach. Not good. And of course, it's still firing on these G cells, which are still making gastrin, which are then coming back and reinforcing some of these activities. So for instance, the gastrin will actually come back and reinforce the production of the chief cells to give you an increasing amount of pepsinogen. Why? Because the idea is the more you have coming in, that is to say, the more food you have coming in, it means the more protein you have, which means you have to make more protein cutter, which is a pepsinogen. So it's the reason why you kind of have this tethered this way, so that you can increase the amount of pepsinogen in response ultimately to the increase in the amount of protein in your stomach. You have more work to do, work harder. That's basically the idea. And then it also increases the parietal cells, because the two of these guys together will basically account for a lot of chemical digestion. You're cutting proteins, the hydrochloric acid is degrading bonds, which it oftentimes does, and gastrin is reinforcing all of that. So this is your chemical side of things. The other thing also that it's doing is it's also stimulating the other plexus, which is the myenteric plexus. So myo for muscle, enteric for digestive system. So these are the muscles of the digestive system. So these are basically stimulated by the myenteric plexus. So these are what's called mixing waves. These are, remember we talked about the washing machine effect, the slosh contraction pattern of the digestive system. You had the smooth peristaltic action. Now you have the segmentation. That's what this one is. This is segmentation. So what you're doing here is you're mixing everything up. And so this represents mechanical digestion. So not only do you have all of the acid and the proteins sort of going to work on the food in a chemical way, but you also now have the stomach kind of doing these sort of kind of choppy segmentation waves, these mixing waves, sort of churning everything up. And what it's basically doing is it's turning what is kind of like a paste. That's the way, the nature of the food bolus as it comes through your esophagus. And ultimately what it wants to do is down here at the pyloric side, it wants to turn it into what's called chyme. So this is more of a liquid. So you go from a paste to a liquid. That's what you're going for. Because if you have the paste going into your small intestines, that's not going to be good. You're going to basically block your small intestines. Right? So what you're doing is basically breaking it down, breaking it down, breaking it down, breaking it down, breaking it down until it gets easier and easier and easier to get access to, so that you can cut up those molecules and absorb them. Right? That's basically what you're doing, is as you're basically liquefying this, you're getting access, more surface area contact, so that you can actually cut these things up. So that's what's happening in your gastric phase. One of the things that I want to mention, though, just sort of as a check, is now notice at this particular point, not only is your food, your stomach filling up with food, right? So the pasty food balls, but you're also upregulating the amount of acid and pepsinogens. All the gastric juices are also increasing as well. So you're filling your tank up, right? And so ultimately, the reason why your stomach doesn't ever pop is because as it distends, it's going to trigger stretch receptors, which will communicate back to the enteric plexuses to say, listen, stop, right? That's it. That's where we stop here. We're already full. We've got a full tank, so stop it right there. And so ultimately, these stretch receptors will communicate in sort of the sensation of satisfaction. So that's the base. Say you go in hungry, you start eating, and then you're not hungry anymore. That's because your stretch receptors are basically telling everybody, it's like, listen, we got food, so you don't have to be hungry. Okay. That hunger, actually, by the way, is stimulated by another hormone called ghrelin, G-R-E-H-L-I-N. So I always call it growlin because it makes your stomach growl, right? So that's the one that initiates your sense of hunger. That hormone goes away once you actually get food in your stomach. But we can still eat more, can't we? And oftentimes we do, especially around the eating holidays. And then, of course, we start to feel that sense of fullness, and it starts to get a little uncomfortable, doesn't it? Well, those are your stretch receptors communicating back. That's what gives you those sensations of satisfaction versus fullness. Okay. Then we basically now move into the intestinal phase. So once we basically done a good job of mixing everything up, we turned it into chyme. Then what we want to do is we want to start to slow down the gastric phase, and then we want to go into the delivery phase. Okay, and that's basically the intestinal phase. A couple of things going on here. First of all, you have to stop your mixing waves. That means you have to take your myenteric plexus and you have to stop it. You have to put the kibosh on it. So you do. And the way that you stop that is by stretch receptors that are in the duodenum. So basically what happens is now that you have chyme, liquefied food, basically, puree, in your pylorus, now what happens is you squirt that liquefied chyme through the pyloric sphincter into the duodenum. And once you do that, what's going to happen, obviously, is you fill that duodenum with the chyme, so it's going to be creating a little bit of water pressure in there, so you're going to feel some distension. And that distension, that stretch in the duodenum, in part, puts an inhibitory signal on the myenteric plexus. It says, listen, we've got work to do now. We got it. Stop churning, right? It's done. You've done your job. Now stop. And so the other thing also is chemoreceptors. In particular, what these guys are looking at is pH, right? Because you go from the stomach chyme, which has a pH of 2, and not only that, but a well-protected, thick, mucus-coated interior lining to protect it from that pH of 2, to a duodenum that does not have that liner, which basically means that one of the first things you have to do is neutralize that pH. Otherwise, you are going to fry up your intestines, and it's going to hurt. You think esophageal reflux hurts, right? Try to get on the business end of stomach acid going the wrong way. That will hurt very, very much. So basically what happens is these guys will sense that initial pH change right in that particular area. So you're talking about right in this area by the pyloric sphincter. This will also put the halt signal on the myenteric plexus. So you stop your mixing. The other thing that also happens, by the way, this is a reflex. You don't have to think about this one, called the gastroenteric or the enterogastric reflex. It's the enterointestines, gastric, putting the halt signal on the gastric phase. The other thing that also happens is you also want to stop peristalsis, because the idea here is peristalsis is the way that you move your chyme through the small intestines. It's also how you deliver the chyme from the stomach to the small intestines, but you want to do this in a regulated way, right? So you don't want the peristalsis like your esophagus just to go without control. You want to squirt it in there when you're ready for it, but hang on to it when you're not. And it's like, okay, I just finished that piece. Now give me some more, right? So you want to squirt it, and then you want to hang on to it. So you want this to be a little bit more controlled and regulated. So you want to have some control over this. So how do we control this? First of all, we got chief cells to deal with, right? Chief cells were busy making pepsinogen, so we got to back that off. Also, the parietal cells were busy making hydrochloric acid, so we got to back those guys off too. So that means we have to have some inhibitory signals on this. And those inhibitory signals come in the way of some proteins that are actually going to be secreted in the duodenum. So basically, once you actually enter your chyme into the duodenum, in that chyme is going to be all the macromolecules of food. So lipids, carbohydrates, proteins, nucleic acid. Now, the presence of lipids and carbohydrates is actually a signal that will cause the duodenum to secrete the hormone cholecystokinin and gastrointestinal or gastroinhibitory peptide, which sounds like what it says, right? It inhibits the gastric activity. Cholecystokinin also does the same thing. So both of these guys will basically inhibit chief and parietal cell activity, and also it will put the halt on peristalsis. So these two are the ones that will put the brakes on all of the gastric phase activities and stop it cold. Now, the decreased pH, thank you chemoreceptors for telling us that the pH is decreasing, right? Because they're the intelligence agency. They're the ones who basically collect all the information. So this decreased pH will trigger the secretion of secretin, which will also work in conjunction with cholecystokinin and gastroinhibitory peptide to also shut down your three main gastro or gastric activities, which is chief cell activity, parietal cell activity, and peristalsis. So a couple of things going on in this intestinal phase is stop the gastric phase, right? That's a big theme there. The other one is deliver the goods, chyme, to the duodenum. So then once we actually work through this, then we have to start talking about not just digestion, which we have chemical and mechanical at work in the stomach, but now we also have to start talking about absorption. Now, is everything perfectly digested once it leaves the stomach? No, there's still a lot of work to do, right? So you're always going to have the theme of digestion as you go through. As a matter of fact, there's some things that will never get digested and just pass them. Just basically, it's they bypass through us and on their way to the toilet. But now what you have for the first time is material that's getting to the point where it's absorbable. So what does absorbable mean? So in order to absorb it, it has to be small. And you have to be able to uptake it through just normal cellular processes. So that means that once you get down to a small enough amount, go ahead and absorb it. There's no reason to wait around. I mean, you're not waiting for the completion of digestion. You got something to absorb it. By all means, absorb it. And so now you start to see that as we start to exit the stomach, we still have the theme of digestion going on. There's still some digestion happening, a lot of it actually happening in the duodenum. But now you're starting to get to that point where you're starting to get a little bit of absorption. So the digestion begins obviously in the stomach with pepsin, right? So this is basically going to be digesting proteins. And proteins, basically, the digestion of proteins is a very arduous process because proteins tend to be very big and they're very folded up. So they're very difficult to get access to. Generally speaking, digesting a protein to completion, to complete amino acids, is a multi-step process involving multiple proteins with specialties, right? So for instance, it starts off with just very generic protein cutters that basically all they do is they just start snipping stuff here and there, kind of like pruning a tree. They're just kind of making the thing smaller. So if you can kind of chop it up and kind of make it a little bit smaller, maybe instead of like a big, 200 amino acid impossibility, now maybe it's only like a 100 amino acid. So you can reduce the size by half. And then there's other proteins that will actually look for very specific patterns of amino acids within that protein, and they'll cut it up in different pieces. So basically what you have is kind of like this progressive sort of chopping up of the protein into smaller and smaller bits until you finally get to the terminal proteins where they cut them up into amino acids right before absorption. So you also have, before this point, you also have the digestion of carbohydrates. So you have salivary amylase in your saliva. It's one of the amino acids, it's one of the enzymes that's being secreted by your saliva. So you're already at work cutting up your carbohydrates while you're chewing. It's one of the reasons why, not just for lubrication to get it down your throat, but also one of the reasons why you saturate your food with your saliva. And then also there's lingual lipase. You do a little bit of lipid work as well in your mouth while you're chewing as well. So those are all happening before we get to this particular point. Now, the stomach itself does not really have a lot of absorption. So the stomach itself is mostly digestion, not a lot of absorption. So there's not a lot of nutrients going on there. So ultimately what's going to happen, what is your goal? For in the stomach, you want your food to become more fluid, turn into chyme, right? This is your liquid. You want your pH to get down to 2. Why? Number one, it basically activates pepsin at 2. So that's one of the reasons why you want to get down to a pH of 2, because it activates your first proteolytic enzyme to start work on those proteins. And you think about it, I mean, your food can be sitting around in your stomach for a little while it's waiting to go into the duodenum. Why not just get to work? I mean, you're sitting here, let's just start chopping you up a little bit. We might make it a little bit easier, move things through a little faster. Your pepsin gets activated, so you get active protein cutter in there. And you kind of start to sort of reduce the size of it. I always like to think of digesting a protein like pruning a tree, like a really, really big tree, right? I mean, you basically start off by kind of just taking off some big stuff. I mean, it's big, it's still pretty unmanageable, but then you get out the chainsaws, these big limbs are on the ground, you kind of start to cut them up into smaller and smaller pieces. And those smaller pieces you cut up into smaller pieces, and you can pretty much put those in the compost bin right at the end of the day. But it's multiple steps, and that's kind of what's happening here. So mostly digestion, not a lot of absorption. So where does the absorption come into play? It begins in earnest in the duodenum, which is the first part of the small intestine. So the small intestines are basically absorptive organ. So this is where you're going to be doing a lot of your absorption. And so the duodenum is going to be about roughly 10 inches long or so. Oftentimes we call this one the mixing bowl. Why? Because it receives the chyme from the stomach, by the way, which is too acidic, right? So that's a problem. Is that me? Not used to getting, must have been some sort of a, okay, anyway. So not only that, but you're going to be taking digestive secretions, adding it in there. I know what that is, but anyway. And also taking secretions from the pancreas and from the liver as well, and mixing it all. So basically what happens is it kind of mixes this into a great mix, like a witch's brew. The wrong witch. Witch's brew. Yeah, kind of like that, you know, boil, trouble and broil, right? Fire, burn, and cauldron boil, right? So that's the only scene I know. That's my favorite, right? And even then, I think I got that one right, because it's not boil. It's like toil, something like that. It's some other word that's in there. But anyway, that was cool, right? I mean, anytime you got witches in a Shakespearean play, you got me. I'll stick around for a few scenes to watch. Or battle, right? Somebody's dying, which I really like Shakespeare, because somebody's always dying. There's only one person who lives. It's a lot of fun trying to figure out who that person is, because it's never the guy you think should live. It's always some schmuck, right? So ultimately then, the job of the duodenum is to sort of take all these secretions from the pancreas, from the liver, and also in the stomach, and mix them all together into this kind of big cocktail, okay, of stuff. Now, one of the things it has to do is to take the chyme from the stomach, which it does, but then it has to get right to work to neutralizing this pH, because otherwise it's going to start damaging the epithelium of the small intestines. And so that's the job of the duodenum, is to basically kind of get this into a workable shape. Then we move into the jejunum, which is like 2.5 meters long. So a meter is roughly about that long. So you got 2.5 of those. So the jejunum is actually quite long. And so ultimately, this is where you get a lot of chemical digestion now that you've sort of mixed it up into this duodenum region, and you start getting a lot of absorption. So this is the main area of absorption. And as a result, you have some what's called plica circularis, which basically is structures. We'll take a look at those structures that increase surface areas, similar to what the rugae were in the stomach. Only they're called plica circularis in the small intestines. And of course, you have villi in this particular case. So this is where most of that absorption is happening. And then, of course, we move into the longest. So instead of 2.5 meters, now we got 3.5 meters. So nearly 4 of those. This, by the way, is 11 and almost 11.5 feet long. Think about this for a second. A basketball hoop regulation is 10 feet. So basically, you can take your ileum from the ground and go 1.5 feet beyond, above a basketball hoop. That's insane. It's just it's all like wadded up in there. It's just like all wound up in there. It's crazy. But yeah, I mean it's really long. So the terminal point where it segues into the colon is the ileocecal valve, which is a sphincter. And of course, this is basically designed to transition the chyme which now hopefully is being more and more depleted of nutrients because you're absorbing it all, segues into the cecum, which is the entry point of the large intestines. Okay. So then let's take a look at a little bit of histology, a little bit of anatomy, and sort of break our important structures together. Because the intestines, really, if you think about it, small intestines, is sort of like the sort of epicenter of your digestive system. This is really where the important stuff happens, where a lot of your absorption happens. So the plica circularis are basically these little folds in the intestinal lining, similar to like the rugae. And they're always there. So you can't stretch them out, right? They're there to increase surface area, like I just said. And then the villi are actually the absorptive organs. They're like finger-like projections that stick out of the mucosa that are lined with your columnar epithelium. And then they also have what's called microvilli on them. So if you take a look at the avillus, what you'll have is basically a finger-like projection of lamina propria that's kind of filling the centerpiece with your capillaries and your lacteals. And then the outside is going to be columnar cells, which are the absorptive cells. So that's basically sticking out into the lumen of the intestines where all those nutrients are as they're passing through your digestive system. And then if you were to take a look at one of these columnar cells, has these like little fine projections of cytoplasm, which are microvilli. It creates kind of like a little hair-like sort of like a grassland almost, especially when you get a lot of these together. And so we actually have a term for this actually in the digestive system. We call this the brush border. It's that little region of microvilli. Now in histology, sometimes you'll see this as sort of like a little dark liner that's right there that surrounds the edge of the epithelium. That kind of little dark line is the microvilli. Okay. And so that again is also to increase surface area. Why? Because the more surface area you have to absorb nutrients over, the more nutrients you're going to absorb, and that's a good thing, right? Because you need to absorb a lot of nutrients. There's also intestinal glands or crypts. So these basically are in between the columnar epithelial cells. So these basically are mucus cells that eject the protein that's associated with mucus, which is mucin. That'll kind of coat those intestinal surfaces. So notice mucus is a theme. It's basically everywhere. That's one thing we make a lot of is mucus. We are slimy little creatures. So open, these basically will be kind of at the base of these villi. You'll see these glands, and we'll take a look at some of these. Some of you guys saw these in your histology. This is also kind of an area where your brush border enzymes will hang out. So in that little grassland, that little microvilli grassland, are enzymes that are called brush border enzymes. And these guys are important because the brush border enzymes are the enzymes that do the last enzymatic cut before you absorb it into your bloodstream. So that's kind of what it looks like. So here you can see a villus, the lamina propria here. So that's basically what this is. That little finger-like connective tissue, that's an extension of your lamina propria. And then at the kind of the valley, if you will, between two villi, you see this kind of like deep pit. That's your intestinal crypt. Okay, so some of you guys saw that. So it's kind of similar to like a gastric pit, only it's more of like for intestinal secretions as well. So here's your muscularis mucosae. This is your submucosa. Notice you've got the vasculature, venous and arterial vasculature, as well as lymph vessels. Those are important because those lymph vessels are necessary for you to be able to absorb your lipids. It's in the submucosa. You notice right here, this nervous piece, that's where your submucosal plexus is. So that plexus is part of the enteric nervous system that's controlling a lot of those gastric phases and things like that, the cephalic phase. That submucosal plexus is in that submucosa of your liner, which is the reason why they call it the submucosal plexus, because it's hanging out in there. Then you can see your muscularis externa with your circular and your longitudinal muscles, and then sandwiched in between your two layers of muscle, your circular and your longitudinal, is the other plexus of your enteric nervous system, which is the myenteric plexus. So the myenteric plexus is basically sandwiched in between the two layers of muscularis externa muscle, and the submucosal plexus is sandwiched in the submucosa. So that's basically where your enteric nervous system control is hanging out. And then, of course, every now and then you might see this kind of like little bean-like structure in the lamina propria, where you see a lot of cells in there. Those are lymph nodules every now and then. So that's kind of like your bodyguard, correct? Your hired bodyguard. So here's a villus, right? Because this is going to be the most important thing. This is your absorptive organ. So here you can see your lamina propria basically poking through the epithelium. So the columnar epithelium is coming up with it. So this is all your lamina propria. And then inside this, you're going to see it's pulling up the blood vessels from both the venous and the arterial to form basically a capillary network. So this is kind of like a vertical capillary bed. A little bit different than the structure of the ones we saw, but still a capillary bed. Also in the middle of it is going to be this green lymph vessel. This is the lacteal. This is the basic of the vessel that absorbs your lipids. So lipids, so now we already have a couple of functions for the lymph system now, don't we? So function number one is obviously an immune function. It's basically the freeway, if you will, for your immune cells. So they basically sort of traffic back and forth from lymph node to lymph node and so forth. Because it's an open system, if you recall, in a capillary bed, we have capillaries, right? We have water going out, water coming in. One of the things that's also in every capillary bed is a lymphatic capillary. Why? Because the lymphatic capillary soaks up all the excess water that doesn't get reabsorbed, right? That's the reason why the lymph system is important in reducing swelling. It's like a sponge. You just sort of sponge up all the excess, and that ultimately will reconnect back and be refed back into the venous system. So you reclaim that water. Number two. Number three is digestively, it's responsible for absorbing lipids. Okay, if you don't have functional lacteals, then you will not be able to digest and absorb lipids, which is not a good thing. And then, of course, you can see you've got nerves going up through here as well. And then if you take a look at your epithelium, you've got your columnar epithelium with your little microvilli here, creating a little brush border. But notice what also you see. You guys saw this histologically as well. You also notice that every now and then you're going to see these goblet cells, basically, which are mucus producers. So ultimately, then what's going to happen is your nutrients are going to be absorbed into this columnar epithelium. Those are the cells whose job it is to absorb your nutrients. And the reason why you create this villus structure is to increase surface area so that you can absorb more nutrients. It's all about surface area. So then how do we move things through the small intestine then? So basically the way we move things through is typically with peristaltic, right? So basically they're slow. Everything slows down in the small intestines because you want to take your time to be able to make sure you get a good digestion, and also in order to get good absorption even more so, right? So you want to be able to absorb all the nutrients. You don't just blast it all through because that doesn't help you, right? You want to be able to go slow enough to be able to absorb everything. And so you have very gentle peristaltic action, just enough to move it through the intestines as it moves slowly toward and into the jejunum. And so what's causing this? myenteric reflexes, right? That's part of the ENS, so we talked a little bit about that. Notice it's not under CNS control, it's under ENS control because it's been delegated. So your central nervous system does not have to think about the slow creep of your chyme through your small intestines, which is a good thing. Also, if you stimulate parasympathetic, you can accelerate your local peristalsis and segmentation, which makes a lot of sense, right? So you do have the ability to upregulate this a little bit if you need to. But for the most part, the CNS is like, just, I'm washing my hands of it. You just do your thing and make sure it goes okay. And I'm going to go work over here on the heart and the lungs and everything else. Okay. So that's basically what it does. But that makes a lot of sense, right? Because what's the nickname for the parasympathetic division of the nerve of this autonomic nervous system? Yeah, it's rest and digest, right? So it makes a lot of sense. So this is it. This is the bullseye of the parasympathetic. So let's take a look at a couple of reflexes. So we looked at the gastroenteric reflex. This is basically reflexes that will trigger changes in motility. Motility just means your movement through the small intestines. That's basically what it means. So the gastroenteric reflex will stimulate motility and secretion. So it'll kind of move things along the small intestine. So that's just kind of like a reflex that just kind of keeps sort of moving peristaltically the chyme from one place to another. Gastroileal reflex, this is the one that will basically relax the ileocecal valve to allow chyme to enter into the large intestines, into the cecum of the large intestines, where you are going to be fundamentally changing your approach to the chyme now at this particular point, because now instead of trying to absorb stuff as you go into the large intestine, what you're going to be trying to do now is largely reclaim all your water, right? So now you got this liquidy chyme, and so what you want to do now is you want to get some of that water back, and you want to start creating and making a much more dense, dry fecal pellet so that you can eliminate. You don't want to eliminate a bunch of water because you'll dehydrate yourself, right? There are, of course, other things that are happening in large intestines. You do get some vitamins being absorbed there, some non-polar lipid-based sorts of things are also being absorbed there, but mostly it's water. You're reclaiming water. So this is kind of what it looks like. So your gastric reflexes, your gastroenteric, this is basically, this is where this is located. So you're going to see from the gastric phase to the enteric phase. And this is going to be changing your kind of transitioning from one to the other. So here you can see this is what they're trying to show here is the distension mechanism, right? So as you get distension, then those stretch receptors are going to kick off that reflex, which is then going to precipitate some of these things. So the gastroenteric is basically moving stuff by peristalsis through the small intestines, all the way through the small intestines. The gastroileal is going to be working specifically right here at the ileocecal valve. Okay. So what about, and of course, then you go into the large intestines where you go into the cecum, and then you start moving through the ascending, the transverse, the descending, the sigmoid, and then of course the rectum. By the time you get through that of the colon histology slide that sort of tipped you off that it's the colon. Yeah, the goblet cells, right? I mean, they're everywhere. I mean, most of the epithelial cells are mostly goblet cells. Why? Because you have to, more so than all the rest of the intestinal system, is you have to grease the large intestines with mucus for lubrication purposes. Why? Because your fecal pellet is getting drier and drier, which means it's going to start getting dislodged in there. It's going to start getting stuck, scraping along the edges of your colon, which is probably not a very great sensation. That would be very painful, right? If it was dry and just sort of scraping its way along your large intestines. So what do? You grease the sleds. Basically, you just put a lot of mucus in there, so it just kind of like comes right out. Okay. Now, lovely. Like that just comes right out, just like the whole performance art thing. Like, stop. You get the idea. Just stop doing it. It's like, we'll forgive you once, but you're doing it over and over again. Stop. Anyway, let's take a look at some of the accessory organs. Now, these guys are not accessory because they're not important. They're accessory because they're not part of the original tube, right? The GI tract is one long tube, and an accessory organ is basically an organ that's not part of that tube. The first, and probably one of the most important ones, is the pancreas. So we already know a lot about the pancreas, right? We already know about the pancreatic acini. We already know that the pancreatic acini is basically full of simple cuboid tissue because we've seen histology slides of it, right? And that they're kind of scattered around our pancreatic islets. Now, the islets themselves, of course, is the endocrine. So this is the exocrine tissue, and the islets are endocrine in nature, which basically means they dump their product into the bloodstream, where exocrine does not. Okay, and so this is a very small portion of pancreas, but of course we all know that the islets are responsible for insulin and glucagon, right? Primarily. So this is kind of what our pancreas looks like. It looks like a Gorton fish fillet. So you can see the tail of the pancreas, the head of the pancreas. The body of it is mostly made of the pancreatic acini, which are busy making pancreatic juices. Or pancreatic fluids, loads those fluids into the pancreatic duct, which then move toward the duodenum, and it splits. So it goes through one main duct, and then there's an accessory duct also that comes out here. So basically, it basically shoots these pancreatic fluids, which are alkaline in nature, because you have to neutralize your pH. And then the other one is it merges, the main duct merges with the common bile duct to mix with bile, which is also alkaline. And then both of these will mix together in a nice little brew, and then they'll basically squirt through this duodenal papilla into the duodenum. Okay, so that's basically your pancreas. So what exactly is it that's coming out of the pancreas? So it's such a big deal. First of all, most of your digestive enzymes are the pancreatic. So up to this point, I mean, pepsin, yeah, it kind of fusses around with the proteins a little bit, but it doesn't get anywhere close. Your salivary amylase, I mean, let's face it, most of us probably don't chew enough. So the less you chew, the less effective your salivary amylase is going to be. So what does that mean? That means you're saving the bulk of your digestion for your pancreatic carbohydrates. So all of your digestive enzymes are coming from your pancreas. What are we talking about? Well, we're talking about things like amylase, right? Pancreatic amylase, which basically goes in there and starts doing work on starches. Pancreatic lipase, which is basically going after lipids. Nucleases, basically, which is chopping up DNA and RNA. It's kind of weird, right? Because most people don't think of themselves as eating DNA and RNA. But guess what? Have you ever eaten a cell before? Yeah, everything you eat is made of cells, and every cell has DNA and RNA in it. So it comes along for the ride. So you also have proteolytic enzymes, basically, which are going after other proteins. Not other proteins, all proteins. So protein, lytic, lysis, to cut. So this is proteolytic. So these are all the protein cutters. It's basically what your pancreas is making. It's making this huge spectrum of all these digestive enzymes, mixing it together with alkaline buffers, and that's your pancreatic secretion. Okay. So pancreatic amylase is a carbohydrate that breaks down starches, very similar to salivary amylase. So it kind of basically picks up where salivary amylase left off. Pancreatic lipase basically will go after lipids, and what it does is it reduces it down into smaller pieces. Doesn't cut it all the way down. It just kind of reduces it down to smaller pieces, smaller fatty acids and things like that that are a little easier to absorb. Nucleic acids are being broken down by nucleases, and proteolytic enzymes are being breaking down proteins. And there's all sorts of different types of proteolytic enzymes. Proteolytic enzyme just basically means a protein that cuts enzymes. I mean, or that cuts proteins. I mean, an enzyme that cuts proteins. There, sorry, I cut that back. So, which is actually, if you're thinking about it, is a protein that also cuts proteins. It's kind of cannibalistic. But anyway, so there are major families of proteolytic enzymes, and they each have a slightly different sort of flavor to them. So there's what we call proteases. So this is a family of proteases. These are guys that go after large protein complexes, basically makes these more simple. So think of a protease as sort of like taking a big old sandwich. A protease is like cutting the sandwich in half or in quarters. So you're taking a big old complex and you're making it smaller to handle. Okay. Peptidases, notice the term peptide, will basically take small proteins and break those down into amino acids. So once you basically get these things down, these big proteins down into small enough pieces, then the peptidases, this is a big family, and lots of them in there. We'll take these protein chunks, if you will, and break these guys down into individual amino acids. Now, when you take a look at what your pancreas produces, this accounts for nearly 3/4 of all of your pancreatic secretions, enzyme-wise anyway, right? So there's going to be a lot of enzyme dedicated to protein, which makes a lot of sense because there's a lot of protein in your food. Even foods that we don't think as protein rich are actually mostly protein, because proteins are the molecule that does most of the work in your cells. So that's the reason why there's so many of them. Generally speaking, these enzymes are important, but they're also dangerous, aren't they? Because they don't care. They don't care if it's your protein or somebody else's protein. If it's a protein, they're going to chop it up. So when you make an enzyme like this and it sees your protein, guess what it's going to do to it? It's going to chop it up. So when you're making hydrolytic enzymes like this, digestive enzymes, you have to be very careful, because otherwise, if you're not careful, you'll self-digest. Bad idea, right? Because it'll go after your own molecules. This is the reason why all enzymes typically are in an inactive state called proenzymes. By the way, whenever you see the suffix -ogen, that is a proenzyme. That's an inactive state. We've already seen that once, right? Like fibrinogen, that's an inactive state. Oftentimes, if you see the term pro on the front, then that is also an inactive state. The idea being, like we saw this also with pepsinogen, right? Pepsinogen is what's made. It has to be activated by acidic pH in order to be able to be activated. And then you do this for a reason, so that you can deliver the enzyme to the site of activity. You don't want an enzyme chewing up your pancreas as it's going down the pancreatic duct. You want it to be inactive until you deliver it. And then once you deliver it, then have a mechanism there to activate it. And then once it gets activated, then you can start tearing up whatever's in there. Okay. Generally speaking, most are activated by pH. Because you're going to see a significant pH change once you go into the duodenum. Okay, so that's the pancreas. Now, what about the liver? If we take a look at the liver, let's take a look at the blood supply of the liver. So basically, when you take a look at arterial blood, about 1/3 of your blood supply is going into the liver. So it's a lot going on there. That's kind of what it does. All of your blood has to go through the liver. It's like customs. Everything has to go through here, right? So basically, this is why those arteries, those hepatic arterioles, are really important. So about 2/3 of your venous blood supply is going through your portal vein. If you think about that. But that's the way the liver is organized, isn't it? Why? Because when you eat food, and this is the reason why we're talking about the liver here, by the way, because when you eat food, where we talked about absorbing your nutrients, right? You absorb amino acids. Where do those amino acids go? Well, we just saw that. In the villus structure, you've got a capillary. So those amino acids go right into the capillary. But where does that capillary go then? That goes into the venous blood system on its way to where? To the liver. and then it goes through those sinusoids and it bathes those hepatocytes. And it's not necessarily feeding the hepatocytes. You know what the hepatocytes are doing? They're metabolizing everything you just ate. So they're making sure that everything you just ate looks good. There's a reason why if you take medication, you always have like a medicine that's out here. It's like, okay, this drug right here should be very therapeutic because it's very active. But guess what? You have to always account for what the liver does to it. Because if you take a medication that looks therapeutic and then it goes to your liver and the liver metabolizes it into nothing, that's pointless, right? Because everything that your body gets bathed in, all the nutrients, comes from the liver first. So if you take a medication and you absorb it into your system, it goes to your liver and you metabolize it into nothing, guess what? You have no therapeutic effect. This is oftentimes one of the reasons why some medications have to be delivered as an injectable, is because you're purposely trying to avoid the liver, because your liver metabolizes it in an unfavorable way, because it has to basically metabolize everything that you put in your mouth goes through your liver. Okay, so your liver is a very, very hard working organ, probably one of the most hardest working organs in your body. So it's basically bringing blood in from the esophagus, from the stomach. If you have anything going on there, small intestines, everything that's absorbing is going into that hepatic portal system. Your large intestines, it's all going into the hepatic portal system. Everything you absorb, all those capillaries that are picking up nutrients are all going to this hepatic portal system into the liver, where it gets screened and metabolized before it moves on to the heart. Because remember, the heart's going to be distributing nutrients throughout the rest of the body. You need to have somebody to make sure that that's safe. It's kind of like, think of the liver as sort of like a mother who's kind of testing out, the baby food before she gives it to the baby. Why? Because she's the one whose job it is to sort of screen everything to make sure it's safe. The baby, once they get it, they're just going to eat it, right? They're just going to, just like the heart. Once the heart gets it, it's just going to grab it and then pass it on to everybody else. So if it's poisonous, guess what? You're just passing on the poison to everybody else, and you're killing yourself, right? So the liver basically is designed to metabolize and to sort of detoxify a lot of those otherwise toxic things that we might eat. Alcohol is a good example of one, right? So basically our liver is broken up into lobules. We remember this from lab, pulled over from lab. And so this is our functional unit. These are these hexagonal areas, which is flanked by these portal triads, which many of us have been working on hard in lab for a little while. So we can do well on our lab exam, which is on Wednesday. Today's Monday, sorry. Right, so your portal triad basically is at each corner. And in this area, you're gonna have your three main pieces, right? So you're gonna have your hepatic portal vein, or at least a venule, right, a branch of the hepatic portal vein. Then you're gonna have an arteriole, a branch of your hepatic arteriole, so you have to add blood to it. And then, of course, your bile duct, all three. So here's what it looks like. This may look familiar. So here you can see all these hepatocytes. So these are individual hepatocytes, and then they are usually around these large cavernous opening chambers. So these are the sinusoids. Of the venous structure. So here you can see there's your portal venule, and basically it's shuttling blood into these little sinusoidal areas, and that blood is basically in communication with those hepatic cells. You can also see your portal arteriole, which is delivering blood to the capillary as well. So it's like a giant capillary bed, and of course your bile duct, which is right next to it. The bile duct basically is collecting bile from, that's what these little green circles are right here. So these like little green circles are bile ducts. So the hepatocytes are making bile, and it's basically putting bile into this bile duct. Why? For digestion. So it's kind of what it looks like. So here you can see the bile being made, and it's being basically dumped into the bile duct from these hepatocytes, where you have the venule, which is also going through these sinusoids, delivering blood to the hepatocytes, as well as the venule delivering blood and sending it through these little sinusoid areas. into what's called these bile canaliculi. Remember that term? We had the similar sort of a situation in bone. Bone was kind of a similar sort of a scenario. You also have a group of macrophages called Kupffer cells, which are sort of there as sort of an outpost, a security detail that basically makes sure that you don't have any bad guys trying to enter the body through this direction. So let's take a look then at the bile production a little bit. So this kind of like is a shot of the gallbladder. So you can see the gallbladder where you have sort of the fundus, the body, and the neck here. But here you can see the actual segments of the, actually, you might want to use this to study also for lab. So this guy right here, this is the cystic duct, which basically fuses with what's called the common hepatic duct. And then the bile basically will go down through the main bile duct, or the common bile duct. So filling these common hepatic ducts is going to be the right and left hepatic duct. So these are coming from the different lobes of the liver as they make their way from these portal triads. Okay, so they're basically draining it down like a tributary system, and they're draining it down into the common hepatic duct. And of course, here you can see your hepatic portal vein. So now let's take a look at this area down here. So as it enters into the duodenal papilla is kind of like this little chamber that you're going to see in the wall of the small intestine. And there's a couple things going on here. So first of all, there's a hepatopancreatic sphincter that's basically governing how secretions are coming through here. And you can see the inlet of the common bile duct and the pancreatic duct. So what's happens is they're going to basically come together right about where the sphincter is. And the bile is going to come through here. The pancreatic fluid is going to come through here. They're going to mix in this little antrum, this duodenal papilla, and then they're basically going to squirt out. They're going to squirt out when this sphincter contracts. So when the sphincter contracts, it's going to basically squirt the secretion that's now mixed inside the duodenal papilla. It's going to squirt it into the duodenum. Very similar to you squeezing at the bulb of a dropper bottle. Okay. So how do we control this then? How do we control this as it's sort of coming through? Because we need the delivery of bile and pancreatic fluid. So especially with bile, how do we do this? So the gallbladder, remember, is basically a reservoir for bile that you haven't used yet. So it's basically a storage container for bile, as well as fresh bile coming from the liver, right? So you basically have fresh bile coming from these two inlets, right? The two hepatic ducts, which fuse to form the common hepatic, and then that fuses with the cystic duct. Now, the cystic duct is taking bile that's been stored and pre-made, so it's already been in there. It's been stored in the gallbladder, and that stored bile is coming through the cystic duct and fusing with the fresh bile that's coming from the common hepatic duct to form the common bile ducts. That's what this guy is here. And then eventually what's going to happen is your bile stream is going to be coming through here through the duodenal papilla, and then it's going to exit into the duodenum, where you're going to be able to digest your lipid droplets. Now, what happens with lipid droplets? So the bile itself is made of alkaline salts. And lipid-like molecules that act like detergents. So what does a detergent do? A detergent basically is like a phospholipid. It's got a polar head and a non-polar tail. Basically what it does. So what it does basically is it takes like an oily spot and inserts its tail in there, and it just basically lifts that spot out. So now you can just rinse that out with water. That's basically how a detergent works. Bile does the same thing. What happens is these like little pro-lipid pieces will basically insert themselves in lipid droplets, and it breaks them up into smaller pieces. This is what's called emulsifiers. So it emulsifies lipids, which basically means is it makes lipids form small drops. Most salad dressings have that in them. Vinaigrettes that you see in the store. And the reason why they have those in there is because you wouldn't want to buy something if it didn't have an emulsifier in it, because it would just have this big glob of oil on top of it, which would look very disgusting. If you've ever made your own vinaigrette at home, you know what I'm talking about. It's just a bunch of oil on the top and it's not very appetizing. And not only that, but it's hard not to get a bowl full of oil when you're trying, like you gotta shake the devil out of the thing, right? So it's like if you don't move quickly, then that oil, then you get just a bunch of oil on your salad, and that's not yummy, right? So that's the reason why most commercial vinaigrettes have emulsifiers in them. There's literally droplets of oil. We can handle that, but not just the big blob of oil coming out. So these are emulsify those fats so that you can get smaller and smaller and smaller so you can absorb them. That's the reason why bile is necessary, is for the absorption of fats. Now, what controls this duodenal papilla, this sphincter, the duodenal papilla? It's cholecystokinin. So cholecystokinin will actually activate the squirt mechanism of the duodenal papilla. It'll also activate the release of bile from the gallbladder. Both of them will be controlled by cholecystokinin. Okay, we already met that one earlier, okay, in the intestinal phase of the digestive system. So when you're talking to look at coordination and control of the digestive system, you both have neural and hormonal mechanisms, a lot of hormonal mechanisms as well. And so you have to coordinate all of these different activities. So let's take a look at some of the neural ones. So ultimately, obviously, this is going to be controlled by parasympathetic nervous system. We have a couple of different reflexes that are associated with it. Sympathetic innervation, by the way, is an inhibitor of the gastrointestinal. So instead of exciting it, you can inhibit it. So if you're basically running from the lion, it's going to inhibit all of these regulations. So you're not going to be digesting at that particular point. You also have your reflexes that we talked about before that are going to be coordinating the movement of chyme along the intestinal tract. So from one place to the other. So what about the hormones? Oh, there's so many hormones, right? There's a lot of hormones here, which we run into several of them. Now what we want to do is we want to unpack those guys a little bit. So they have lots of different effects. Most of them are essentially designed to guide the activities of the stomach. So these guys are our list right now. So we ran into gastrin, secretin, gastroinhibitory peptide, cholecystokinin. We haven't seen vasointestinal peptide. We'll talk about that one in a second. And endocrine, we'll also talk about that one. So let's start. Gastrin. So this one is the one that's produced by the G cells, right? We've already seen this one. And what it does is it basically increases stomach motility, right? So stomach activity, and of course, enzyme production. That's pepsin, for instance, if you're keeping track. So it's basically involved in segmentation and the enzyme. So if you recall back when we looked at the intestinal phase, remember gastrin was in charge of stimulating both the chief cells, right? That's pepsinogen production, the parietal cells, which is acid production, and the mixing ways, the myenteric plexus, which is the segmentation, right? That's sloshing and churning and mixing. So that's what gastrin is doing. Secretin is basically stimulated when chyme enters into the duodenum. And so what it does is it works along with cholecystokinin. It increases the secretion of bile and its buffers by the liver and the pancreas. So this is basically the secretion. So it basically is a making more. So more of those buffers. So it's designed to stimulate the production of bile and pancreas. And we'll take a look at that with respect to cholecystokinin, because in cholecystokinin, and I'll loop back around with gastric inhibitory protein. But in cholecystokinin, you're looking at pancreatic production and the enzyme. So basically what happens is the combination of cholecystokinin and secretin are overlapping but complementary. So one is just an upregulation for production, that's secretin. But cholecystokinin both does production and secretion of digestive enzymes, but the sphincter, the hepatopancreatic sphincter, the one that's associated with the duodenal papilla, that's the one that gets relaxed, allowing bile and pancreatic juice to be injected into the duodenum. So secretin upregulates the production of bile and pancreatic juices. Cholecystokinin does the same thing, but it also then initiates the delivery. Secretin by itself will not allow you to get the bile and the pancreatic juices into the duodenum. That's called cystokinin. So you have basically a supply and a delivery, right? So cholecystokinin allows for the delivery of your supply. Secretin and cholecystokinin together work as a team to increase supply of bile and pancreatic juices. Notice that this also happens when your lipids and partially digested proteins, thank you, pepsin, enter into the duodenum. These are the signals. These are the triggers. So the two of those work together. Gastroinhibitory peptide, basically this is happening when you have fats and carbohydrates in the small intestine. So this inhibits gastric activity. So you turn that phase off as you move into the intestinal phase of your digestive system. Vasointestinal peptide, this is an important one because it's going to increase your intestinal gland capabilities, but it's a dilator. So it dilates capillaries to account for and to allow for more absorption. So you also inhibit acid production. That is basically, it's inhibitory to the parietal cells in the stomach. So again, notice an intestinally active hormone shutting down the gastric activities and also dilating capillaries so that you can get maximum absorption. So you can absorb more nutrients into your cell. And then last but not least, enterocrinin is basically when chyme just enters the small intestines, probably stretch receptors, but this one is going to be producing mucin in the duodenum. Basically, this is greasing the tracts. So you're going to have stuff moving through. So you increase the mucus production to grease the sleds so that all this material can move through efficiently and easily. It's kind of like lubricating the chain on a bike. You want to make sure that it's moving smoothly and that everything's going well. Okay, this is kind of what it looks like when you sort of put it together in a flow chart. So here you can see gastrin at work in the stomach itself. So you have acid production, and you also have the myenteric wave mixing, where we have both chemical and mechanical being controlled by gastrin. Gastric inhibitory peptide inhibits that one. This is going to be happening and turning on in the duodenum. Secretin and cholecystokinin work together because these guys are going to be stimulating the production of bile and pancreatic juices, and cholecystokinin allows for the delivery of that. And of course, inhibitory peptide is dilation so that you can actually facilitate nutrient absorption. Once you get nutrient absorption, then basically you get everything that you need in your blood in order to feed your tissues. Okay. So this is kind of a nice little flow chart that kind of puts all the hormones together, or most of them anyway. And of course, you have enterocrinin greasing the sleds to make sure everything moves along nicely and doesn't get stuck. So you have digestive enzymes being produced by your salivary glands, right? You actually have lingual lipase. Lingual, your tongue actually produces lingual lipase. Your stomach is producing pepsinogen. Your pancreas is producing most of it, everything else, pretty much. So you have several different organs that are basically making these digestive enzymes, many of which are designed to break down different macromolecules, which you learned about in general bio, right? Through our process of hydrolysis. Hydro, which means water. Lysis, which means cutting. So you're cutting these things apart with water. And so you have carbohydrases, which break down your carbohydrates, proteases, which are going after proteins, lipases, which are going after lipids. And then, of course, proteolytic enzymes, which are going after proteins. So that's all happening in your chyme. That's all happening in your duodenum as it mixes all those pancreatic fluids as they get delivered into the duodenum. So it that's those alkaline buffers of the bile and the pancreatic buffers mix with the chyme, which is now P acidic, neutralizes the acid, activates all those enzymes that came with the pancreatic buffers. And so you start activating all those protein cutters, and they start going after the proteins, and the carbohydrates start going after carbohydrates, and everybody's starting to get active. And then by the time you get down to the jejunum, you've got a lot of cutting. And now you're sort of down to this point where you maybe have small chunks, very small chunks, like disaccharides, for instance, in the sugar department, phosphates, things like that. So nucleotides are being broken down, but now you have to break down the nucleotides. You can break those down into sugars and the phosphates and the nitrogens. And these brush border enzymes, these guys are the last cutters. Before absorption. So this is kind of what it looks like. So this is basically a couple of different enzymes that you can see here, and it tells you where they are. So pancreatic alpha amylase is in the pancreas, salivary amylase is in the salivary glands. And then at the very end of it, so you're kind of like as you move through your digestive system, you're moving up, right? So your salivary amylase is the one that gets the first shot at it. And then it goes into your stomach. It's put on hold as everything gets denatured, and then in your small intestines, your pancreatic amylase will pick it back up. And then at the very end, your brush border enzymes, maltase, sucrase, lactase, will do the final cut, and will turn these things into monosaccharides, glucose, things like that, fructose, galactose, and then you'll get absorption of your carbohydrates. So this is what a carbohydrate absorption pattern looks like. So you can see your salivary amylase will start the work, and then once it goes to your esophagus, nothing happens. Your salivary amylase gets denatured in your stomach because it's a pH 2. Everything gets denatured there. So now what you have from what you have in your mouth is varying amounts of disaccharides and trisaccharides. So you do a little bit of cutting, just not a lot, right? You just kind of got the ball rolling. And then once your chyme enters into these small intestines, your pancreatic amylase really goes to town on it. So now you're converting most of your starchy sugars that you ate into disaccharides and trisaccharides. So that's three sugars versus 2 sugars. And then once you move into the jejunum area, then those brush border enzymes are going to take these disaccharides, cut them into monosaccharides, and that's what you're going to absorb into your capillaries. Okay. Or it'll take your trisaccharides or whatever, and then these brush border enzymes will do those final cuts, and then they'll take them into your columnar cell. Notice this is a columnar cell here. And so it'll take them into the columnar cell, and that columnar cell will basically take it in and then basically allow it to absorb into the capillaries. So that's how you absorb your. So these are the proteases. Notice there's a lot of them. You don't need to know them all. Notice we have some similar friends here. So here's pepsinogen. So there's pepsinogen, a couple of pancreatic ones that are really big, chymotrypsin, trypsin, those are all pancreatic, carboxypeptidase. So these are all pancreatic enzymes that the pancreas is producing once it goes back in there. At the brush border, lurking in the weeds, so to speak, those final cutters, we have dipeptidases, peptidases, and enteropeptidases. So these are the last ones. You also have another one, renin, 2 N's, not 1, not to be confused with the renin of the RAS system. Right, this is an enzyme that will go after milk proteins. This is the enzyme that we usually use to make cheese with. Right, it's usually like an intestinal protein, but it'll like curdle the milk. So it'll denature the milk proteins, and then that's kind of what gets those curds happening, and then we'll be able to separate that out. So most commercial cheese making is probably using some renin to curdle that. Unless you want like vegan cheese, which I never did understand, because it's like, okay, wait a minute, is it really vegan if it's like, no? But anyway, but sometimes people will make cheese with lemon juice, and that's fine. You can do the same thing that way. So, so this is basically a lot of your enzymes. So here you can see you kind of start the process up here with lingual lipase. And then nothing happens for a long time as you go through the stomach. You get neutralized in the small intestines. You have some, excuse me, these are lipids. You have some, notice fats have been broken down into like fatty acids, like little small fatty acids and things like that. But generally speaking, it diffuses and goes into the cell. So there's no brush borders in here for this one. So typically you take these monoglycerides and fatty acids, and then you'll build them into triglycerides. Remember triglyceride? It was your fat molecule, right? It's 3 fatty acids connected to a glycerol backbone, right? So that's what you're making there. And what they basically do is you load those triglycerides onto a protein complex called the chylomicron. This is a fat carrying protein. And this is how we carry our lipids, right? We have what's called lipoproteins, which are basically designed to carry lipids. And so in this case, a chylomicron is like a big complex protein whose job it is to grab onto these triglycerides that you just consumed and you built. And it kind of carries them in a nice convenient package. And they themselves, when they're carrying the lipids, these triglycerides, they're soluble in your blood. So they can move around in your blood. So that's the reason why you use chylomicrons, is to basically allows your lipids to be soluble in blood. By the way, this shouldn't be news to you. We've run into this many times before, haven't we? This is the reason why you have shuttle proteins like albumin and transthyretin and things like that, and TBG, to move your lipid T3 and T4 around. This is the reason why we have so many shuttle proteins as a component of our plasma protein complement, is because we've got all these lipids to run around in our system, right? So this is basically what these guys do. This happens to be the protein whose job it is at the lacteal to absorb the triglycerides that you eat. Okay, so that's its job. So from here, it'll move through exocytosis into your lacteals. Notice, not into your bloodstream, into your lymph system. And so that was the way that it will actually enter into your body, is your lacteals will absorb your fats. Okay. And then of course, here's our lingual lipase and our pancreatic lipase. And then of course we have our nucleases. So here's our proteins, got that one out of sync a little bit. But here's our proteins. So our proteins, we're not doing much at the beginning. Right, our pepsin gets kicked off in our stomach in earnest. And then as we get into our pancreas, that's when we really unload. I mean, we just unload both barrels on proteins in our pancreas. And then here we basically create these short little peptide chunks and individual amino acids. And then from here, that our little dipeptidases, these are our brush border enzymes, are kind of lurking here in the weeds. And they'll make those final cuts. So if it's a dipeptide, then it'll cut it into an amino acid and take it in. And then it'll absorb that amino acid into the bloodstream. Okay, we'll make that last little cut. Not unlike we saw with the carbohydrates, right? So we had those like little brush border enzymes for the carbohydrates kind of making that final cut. So instead of being a disaccharide, it's a monosaccharide, and then you absorb that into your bloodstream. Ooh. And that's the business end of it. And once you've done that, now you're ready for the urinary system. So let's take a look at the urinary system. So a couple of things that I want to go over quickly, because I know we talked a little bit about the structure in lab, so I don't want to do too much duplication. But the functional piece of the urinary system is the nephron. Right, so here you can see basically you've got the, which is basically a modified capillary. So the glomerulus, which is this capillary here, this guy right there, that's basically the capillary. It's got an inlet, the afferent arteriole, which I basically think of as a rival, and an efferent arteriole, which is the exit. So this is the exit arteriole, and sort of while it's in there, it basically breaks up into this large capillary. And now around it, you're going to have this tubular system. right? And so the tubular system begins with what's called Bowman's capsule. So Bowman's capsule is sort of like this large purple thing, which kind of wraps around it. And if you recall before, where we talked about the heart and pericardial sac, we said just take a balloon and put your fist into it. The exact same thing happens here. So if you take the glomerulus, like your fist, and just put it into a balloon, that's exactly what's happening. So you have like a visceral layer laying on top of the glomerulus, and you have a space, and you have sort of the outside of the actual capsule itself. So that's kind of what you're seeing here. So the capsule is actually has a visceral layer that's resting on top of the glomerulus. There's a space, and then there's an outer piece of it, and then it goes into a couple of pieces of the tubular system. The first part is what's called the proximal convoluted tubule, where you have a lot of absorption of water and secretion of different sorts of things. And then it goes into the nephron loop, where you have a descending limb, you have the actual tip of the loop, and you have an ascending limb. And typically what's happening here is you have the reabsorption of water. So you're reclaiming a lot of water back into your tissues so that you don't basically just dump all that out into your urine. And then ultimately from your loop of Henle, you're going to get to your distal convoluted tubule, which is right here. Again, you're also basically secreting and dumping a lot of different things, either where you're making your final adjustments, and then you go off into your collecting duct. We're going to take basically a stepwise journey through this system, starting first of all with the glomerulus. So the glomerulus, you can see, is basically this big long guy right here. So you can see your afferent arteriole moving inward. It's going to be delivering blood into the capillary itself, and then it's going to be going through this capillary system, and then it's going to be coming out through the efferent. Now, typically smashed right in this area is a piece of the distal convoluted tubule. So the distal convoluted tubule actually kind of bends back around and essentially kind of smashes right there by where the glomerulus is. And this is important because this little wedge right here is what's called the JG complex, the juxtaglomerular complex. The cells of the distal convoluted tubule, which tend to be a little more compacted, and you guys see this in your histology, is what's called the macula densa. So this is a sense, this senses sodium. And that little triangular wedge of cells that kind of gets trapped in there from the distal convoluted tubule, these guys are the juxtaglomerular cells. These guys are regulating and monitoring blood pressure. Now, this is perfect, right? Because this basically allows you to be able to monitor blood pressure as it's coming in to the system. So basically, as it comes into the system, you want to make sure that your blood pressure is just right. Remember, we talked about getting the blood pressure and the blood flow just right in a capillary. You have to have enough blood flow to get good exchange, but not so much that you blow up the capillary. Same thing's also true here. So this is one of the reasons why you have your main blood pressure sensors, the JG cells, right there at the doorstep of the blood as it's entering. So you can think of this as sort of like the proverbial doorman of the club, right? Checking you down, making sure that your blood pressure and your blood flow is sufficient. Now, once it basically goes in, you can see basically the pieces of the glomerular capsule. So here's the space right here. This is the outer wall of the glomerular capsule. And these little guys right here, these are what's called the podocytes, or the visceral epithelium. So what they basically do is they do something that's a little interesting, is instead of just sort of resting on top of the glomerulus, what they do is they kind of wrap themselves around the capillaries, and they interlace like your fingers in between each other. And they create, this is what creates slits. We call these filtration slits. So these cells basically create these sort of finger-like projections, which is called a podocyte. So podo, foot, site, cell. This really is referred to as a foot cell. That's what this is referred to. And so these little sort of fake feet, or these fake false feet, basically interweave between each other, and they create essentially what's kind of like a sieve-like structure, right? So it's like sifting sand between your fingers. So if you basically take a bunch of sand and you sort of let it sift between your fingers, you're doing exactly what these podocyte cells, these capsule cells, are doing. Okay, now this is important because this forms a filter. It's almost like a sieve type of an effect, which is interesting because one of the things you want to do is you want to make a filtrate that is filtered out, right? So that's basically what you're doing. You're filtering out your blood. And so we want to take a look at the process of glomerular filtration. Now, ultimately, what we want to talk about, just like we had a respiratory membrane in the respiratory chapter, we have a filtration membrane in the urinary chapter. And just like before, it's a thin membrane. So the first thing is going to be your capillary endothelium. Then is going to be a region of connective tissue called the dense layer. And then you're going to have these filtration slits. Look something like that. So here's the inside of your capillary. And this would be the capsular space. So the idea is you want to push your blood plasma through this filtration slit into the capsular space. So let's take a look at it. So the capillaries, first of all, this should sound reminiscent, right? Because all the capillary physiology is the same pretty much. In this case, we're going to be running into fenestrated capillaries, which means what? That basically means the fenestrations themselves have a diameter to them, and the diameter is between 60 and 100 nanometers. It basically means you can let things that are about 60, 100 nanometers pass through these. So no blood cells, sorry. You can get some solutes and small plasma proteins, but no large proteins. Can't do that, right? So this is basically a sieve. So the capillary itself is a sieve, and it's part of a sieve-like structure. So you got like a double sieve, like a double sifting, if you will. So let's take a look at the dense layer. The dense layer itself has some more selection to it. It's more restrictive. To allow small plasma proteins, nutrients, and ions, right? So things like glucose will get through, things like that. Ions, sodium, calcium, stuff like that. Very, very tiny stuff. And then the filtration slits, believe it or not, are actually the smallest. So this is the smallest filter. It's now down to 6 to 9 nanometers. So you started off at 60 to 100 nanometers. So these are large holes. And then you started going down progressively to 6 to 9 nanometers. So this is a very small hole. So that's the effect. It's almost like a funnel. A lot of stuff can get through the filtration for the fenestrations, right? Because they're 60 to 100 nanometers. But then more things are going to get stopped at the dense layer. So only the small, like a lot of the bigger plasma proteins that got through the fenestrations are going to get stuck in the dense layer. And now only really tiny things are going to be able to get through this particular last passage, right? So very small, tiny proteins. And we just keep saying small. What we're talking about is smaller. It's a progressive smallization, if you will. That's a term of these plasma proteins. And so you can see it getting smaller and smaller and smaller. And so here's the fenestrations of the hole. So some things will make it through here, but then they'll get stuck in the dense layer, and then they can't get past that. And some things will make it through the dense layer, but then they'll get stuck in the fenestrations. They can't make it there. And then only the smallest things will make it through the pores, through the dense layer, and then all the way through the fenestrations, where they find themselves into the capillary space. And they have now officially become part of what's called the filtrate. So the filtrate is like preliminary urine. It's mostly plasma-like at this point. So what you're doing is you're taking your plasma as it's going through this glomerulus, you're taking your plasma and you're pushing it against this sieve. And basically all of the small stuff in your plasma is basically pushing through the sieve, oozing through the sieve, and it goes into the capsular space. And so the capsular space then is mostly looking like plasma at this point. Obviously, though, urine does not look like plasma. So we're going to have a lot of alterations to do to this filtrate in order to get it to look like urine. It's not there yet. Okay, that's the story of the tubular system, is modifying this filtrate to look more like urine. So let's take a look at the filtration idea. Because ultimately what you have is you have a blood pressure, right, that coming into the glomerulus, and you're pushing this against your filtration membrane. And so it sounds very reminiscent, doesn't it? Wasn't that sound like a lot like we talked about in the capillaries, where basically you're pushing that capillary hydrostatic pressure against the capillary, getting filtration at the beginning? Well, you're doing the same thing, only in this case, you have hydrostatic pressure at the beginning that's pushing against this filtration membrane. That's fluid pressure. And then, of course, you have your colloid osmotic pressure, which is the amount of the solution, the dissolvables, if you will, that are causing the sucking effect, right? So this is the sucking effect on water. And that's on either side of the capillary, because you got dissolved stuff inside and outside, right, in the capsular space as well. So it's a dance between the two of these. Take a look at hydrostatic pressure. You have what's called glomerular hydrostatic pressure. This is basically the blood pressure inside the glomerulus, right? Just like capillary hydrostatic pressure was the blood pressure inside the capillary, glomerular hydrostatic pressure is basically the blood pressure that's in the glomerulus. It's coming through the afferent arteriole. Now, this basically will have a strong push. It'll tend to push out of the plasma and into the filtrate. So this is an outward push, generally speaking. And it's generally a higher pressure than in capillaries. Why? Because of the arterioles. They're resistance vessels, so you can sort of mess around with that a little bit at this level. Then ultimately, the blood as it exits is going to go through the efferent arteriole, typically with a smaller diameter to maintain forward blood flow. And it also will produce resistance, so you can basically increase the blood pressures to force the blood into it. This is the nice thing about the efferent afferent. Because they're arterioles, they're resistance vessels, you can regulate what's with the blood pressure inside the glomerulus. So if you want to increase the blood pressure in the glomerulus, basically constrict the efferent, that's going to bottle everything up. It's going to be higher pressure, and it's going to basically build up that pressure in the glomerulus to make sure that you get good push against that filtration membrane. Or if you want to increase blood flow, you dilate it and you allow blood flow to go through. And you can do the same thing on the afferent side of it, right? If you want to back off on that blood pressure, then you can dilate. And so you can kind of play these games of dilation and constriction on either side to maintain and regulate blood flow. And that's a critical task that's actually happening. There's also capillary hydrostatic pressure. What is this? This is the water pressure of the capsule. So the water pressure of the capsule pushing back into the glomerulus is what capillary hydrostatic pressure is. And so this is going to want to go out of the filter and into the plasma. So this is an inward push of water. And so as a result, this is like your headwind. So this is your resistance to your filtration. This resists filtration. Generally speaking, this will usually be fairly consistent. It's usually on average around 15 millimeters of mercury. So that's a fairly stable amount, which is good because you can change up the glomerular hydrostatic pressure, but generally speaking, your capsular hydrostatic pressure is usually fairly much the same. So then when we take a look at glomerular filtration, very similar to what we saw with the capillary as well, it's the net hydrostatic pressure versus your colloid osmotic pressure. Right, so you have your net of glomerular going in, or going out rather, into the filtrate versus your capsular wanting to go the opposite direction. Generally speaking, you make sure that this one is very high and that this one is fairly constant. The osmotic pressure, so as you push fluid out, then you're going to be increasing your blood colloid osmotic pressure. This will draw the water back out of the filtrate. So this is an inward force. So that opposes filtration. And that's averaging around 25 millimeters of mercury. Okay, so there's an inward force, a couple of inward forces that you have to push against, right? So this results into resistance. This is like a resistance thing. So when you're trying to push this stuff across your filtration membrane, these are the resistances that are coming at you that you have to overcome. Similar to the way that the left ventricle had to overcome the overall systemic resistance to get forward movement. The same thing's kind of true here. So then, what is it then? What is your actual net push forward? That's what we call your net filtration pressure, your NFP. Ultimately, it's the net of the inward versus the outward. So ultimately then, this is the pressure that essentially forces things out of the glomerular capillaries into the capillary space, basically making filtrate. And it's the difference between your hydrostatics and your osmotics. So let's take a look at and see what this looks like. So first of all, the big one is our glomerular hydrostatic pressure, right? This is basically our blood pressure. So this is pushing out like a lot. So there's a lot of force here, right? 50 millimeters of mercury is what we're looking at. The pushback, right? These are inward forces, our blood colloid osmotic pressure. That is the sucking effect of our own plasma proteins and blood cells in the blood, sucking that water back in. That's coming in at around 25 millimeters of mercury. The hydrostatic pressure of the capsule is wanting the water to go in by 15 millimeters of mercury. So if you kind of do the math, that balances out. There's an overall net direction, a net filtration pressure of 10 moving outwards, right? So once you basically subtract all of your opposing forces minus your push force, then your leftover is your net filtration pressure. The same thing that we had, basically the same exact idea that we had when we looked at capillaries before. Okay. Okay, so that gets us to our GFR, which is basically our glomerular filtration rate. And this is the amount that your kidneys can produce, that is the amount of filtrate your kidneys to produce per minute. On average, you're talking about 125 milliliters a minute. It's a lot. That's what you guys have been doing for the last 2 hours right now as you're sitting here. Right? So about 10% of that is going to be delivered to the kidneys. So it leaves the blood space, enters the capillary spaces, and then that's kind of what we get in terms of filtrate. So here's the problem. First of all, when we're producing filtrate, we're generating, a glomerulus would generate roughly about 180 liters of filtrate a day. Think about that for a second. A liter, how much is a liter? Roughly about, right, like about a liter bottle or a 2-liter bottle of soda. You have 180 of those per day. Are you urinating all of that per day? Like not even close, right? So obviously, even though you're making the filtrate, you have the benefit of cleaning that up, sending that through that filtration membrane. So you kind of clean some stuff up a little bit. Obviously, you're going to have to reclaim some of that. So most of that is going to be reabsorbed. Because it would be catastrophic, by the way, if you were letting go of 180 liters of filtrate per day. What that means is you would have to be drinking water almost nonstop to replace those fluids. Yeah. So in terms of our clearance and our filtrate production and our ability to sort of move solutes through, you probably have heard of clearance tests. So we have a series of clearance tests that you can take to make sure that your glomerular filtration rate is working well. Typically, you do this when somebody suspects kidney damage or nephritis or something of that nature, compromise in nephron production. And there's different things you can take. You can take creatinine. And basically what will happen is you kind of charge yourself up and you just allow it to clear out of your kidneys. And the amount of time it takes to clear out of your kidneys, they can calculate or they can measure, and they can compare it to how normal a normal kidney function would clear that out. The problem is creatinine is one of our metabolites, so we can't actually metabolize it, which would skew the results. But inulin is one that we don't metabolize. So if we give us inulin, then we'll just basically pass it on through, and then they can measure how well we're passing that through to determine how well we're producing filtrate. Okay, so a urologist would do something like that, or an internal medicine doctor if they suspect kidney failure or kidney problems or something like that. So our filtration pressure, our rate basically depends on our pressure. So how fast we're making filtrate depends on the pressure itself. That is the glomerular filtration pressure. So that'll increase our glomerular filtration rate as well. So we have basically, obviously, levels of control. Autoregulation, which is typically how you're regulating things closely. Hormonal, and of course, nervous. So this is actually a good place to begin on Wednesday. And we should have a fighting chance of getting this chapter completed on Wednesday. So, which reminds me, I still have a lot of our other chapters to post. I think I have at least 2 recordings to post for you guys. So